Um, Welcome, everybody. Thank you so much for joining us. Welcome to the Melanoma Research Foundation's educational webinar this evening. My name is Sarah Seelig. I'm the founder and director of the CureOM initiative at the Melanoma Research Foundation, and I'm joined here tonight by some esteemed colleagues. And the first of which I will introduce you to is Miriam Kadosh, the Director of Education and Patient Engagement at the Melanoma Research Foundation. So Miriam, I will hand it over to you to say hello first this evening.
Thank you, Sarah. It is so great to see so many of you on this webinar this evening. I hope you all had a wonderful Thanksgiving. It is so great to see you, and we hope to continue to provide meaningful educational and supportive opportunities for you and the OM community. So please do not hesitate to reach out to me. My email is cureom@melanoma.org. You can reach me there. And I want to share some upcoming programming that might be of interest to you. Just real quick before we get the program started, we have 2 ocular melanoma support groups that run the first one the first Wednesday of the month and the 4th Thursday of the month. I hope you got a chance to interact with us this month on social media, as this November is our #IGetDilated campaign. Please know that we do post content throughout the whole year, and there will always be opportunities posted online to help you get involved. And lastly, I want to remind you to save the date for our ocular melanoma patient meeting, which will be taking place in Boston, Massachusetts this upcoming year. The weekend is May 2nd through 5th. In Boston. So we hope to see you there. We will be sending out more information shortly, so look out for that. And if you don't see it, please feel free to reach out to me again at cureom@melanoma.org. Lastly, I just want to say that at the end of the program this evening, there will be an evaluation that pops up on your screen. Please do fill that out for us. It really helps us as you provide us your feedback and thoughts. It's so important to us as we plan for future opportunities. All right, I guess it's time to get on with the show. Sarah, I'll turn it over to you.
Thank you, Miriam. So again, we're pleased to have everybody join us tonight for this educational opportunity entitled Creating a Shared Vision of Progress and Hope. And the goal today is to hear and discuss what researchers were discussing at the recent CureOM Global Science Meeting in Philadelphia earlier this month. So as a reminder, the mission at the Melanoma Research Foundation is to eradicate melanoma by accelerating research while educating to and advocating for the melanoma community. We know that education is critical for patients to make informed decisions about their care. So a bit of housekeeping before we get the rest of the program going. We encourage everyone to use the Q&A box to ask questions throughout the session. We'll have Q&A time at the end. And a reminder that the information presented during today's session is for educational purposes, and any individual treatment questions should be directed to your healthcare provider. We encourage everyone to visit melanoma.org and cureom.org to learn more about the resources we have available. And also, this session tonight will be available for viewing later as part of our videos on demand library. So you can share that with others or rewatch this, or if you miss something, don't worry, you'll be able to review the information again. So as most of you hopefully know, tonight we are joined by Dr. Marlana Orloff, Dr. Marcus Butler, and Nigel Deacon. And so I'm actually going to start with Nigel tonight. And we're so grateful to you, Nigel, for being here with us this evening. Many of you probably know Nigel, who is a retired educator and lives in Victoria, British Columbia, Canada. Nigel was diagnosed with ocular melanoma in 2010 and metastasis in 2012. He went on to found Ocumel Canada with Kathy Barnard, the founder of Save Your Skin Foundation in Canada, with the goal of supporting and advocating for ocular melanoma patients across Canada. We thought that we would open with Nigel and maybe some of my own reflections tonight, just as a patient and family member sort of in the room at this global science meeting, listening to the conversations and the research updates. And I think from my own perspective, thinking back to when my husband Greg was fighting ocular melanoma, it was just so amazing for me to be in the room and to hear how the conversations have changed with ocular melanoma and how many research studies are ongoing and how many clinical trials there are and how many options there are for patients now and how that has really evolved over the past several years, the past decade for sure, and how much that has been accelerated the past few years. And so Nigel, I wonder how for you, as a patient sitting in the science meeting, how the meeting felt to you overall, and kind of what kind of perspective did it leave you with?
Thanks, Sarah, and thank you for the invitation to speak tonight. The meeting in Philadelphia on November the 6th was an amazing experience for me. I was aware that you had been working for, I think, perhaps 11 years in building that event. And so I was surprised to find an enormous room full of people from the US, Canada, Britain, France, and perhaps some other places as well. The format was busy, to say the least, and I loved the way that there was a platform for basic researchers working in the field, and the translation then to clinical practice. I was incredibly excited to hear about the clinical trials that are coming soon to a space near patients. And then, of course, the industry partners. I mean, some of the therapies are not clinical trials. They're things like Delcath and Trisalus. Those companies were there to talk about what they are providing to the community as well. So yes, in 2010, when I was diagnosed with primary disease, and even more so in 2012, it was really a grim picture. There was very little available by way of help. And it was extraordinary to see all of these people, discussing their contributions and sharing their experience. In the old days, when you did science, you tried to keep it quiet so you would have all the glory to yourself. That seems in the distant past now. The sharing was an amazing experience.
Thank you, Nigel, for those reflections. I really appreciate them, and I second them. I think the dedication and the commitment to collaboration of the clinicians and researchers in the ocular melanoma field right now, I think is so inspiring. So I really appreciate that. And Nigel, I know we'll get a chance to hear more from you later in the webinar tonight as well. But let's hear from a couple of those inspiring and dedicated clinician researchers tonight. We're so grateful to Dr. Marlana Orloff and Dr. Marcus Butler for being here with us tonight. I think you are two people who need no introduction in this space. So I will just briefly mention Dr. Marlana Orloff is a medical oncologist at Thomas Jefferson University Hospital and the Sidney Kimmel Cancer Center in Philadelphia, Pennsylvania, and grateful to Dr. Orloff for talking to us about some of what was discussed at the Cure-OM Science meeting earlier this month. And Dr. Marcus Butler, who will follow Dr. Orloff, is also a medical oncologist at the University of Toronto. And we're just grateful to both of you for your presence at the meeting and your contributions that day. You both led sessions and participated. actively in the discussion, and we're grateful for you joining us tonight as we report out to the patient community what was discussed at the meeting. So Dr. Orloff, I'll hand it over to you to get us started on this component.
Okay, all right. Hi, everyone. It's always, I can see there's 48 participants. I can see your names. I can't see your faces, but it is nice seeing everyone tonight. So I'm going to go ahead and share my screen. Okay. All right. How's this look? We up?
Good.
Okay. Yep. All right. So, you know, before I get started, I just want to highlight again, you know, some of what Sarah and Nigel have already talked about. I mean, we titled this webinar, Creating a Shared Vision of Progress and Hope. And if you were in the room at that meeting a few weeks ago, that is certainly what was happening. I've been to a lot of science meetings, a lot of uveal melanoma meetings, and I think it was, you know, a lot of folks were in agreement, and we discussed this after this meeting, that it was probably one of the most inspirational meetings that we had been to in a long time. I mean, it was a packed day with just, you know, kind of advancement after advancement after advancement. And the advancements really are kind of spanning the breadth of, you know, primary eye treatment all the way to metastatic disease treatment. So it truly was an inspirational day, and very happy to be here to share some bits of it. So I'm how we're going to do this is I'm going to take kind of part of it to summarize, and then Dr. Butler will take the other half. The interesting thing about the meeting this year is that we, in addition to inviting industry to kind of sponsor the event, we actually invited a number of our industry partners to really participate and actually have their own time to present to the group. And they were really tasked with, you know, giving us updates on their, you know, company, their pipeline, what they were working on in the uveal melanoma space, and then really also just opening it up for discussion about how, you know, we as a clinician community, certainly how the patient community can help, you know, to see some of these treatments get FDA approved and so on. So I do want to note, and I think Sarah might have mentioned this, but this year Society for Melanoma Research actually marked its 20th anniversary, and really 10 years since it was actually held in Philadelphia. And I actually started the introduction at the Cura meeting talking about that the first ever SMR that I had been to was actually 10 years ago in Philadelphia at the 2013 meeting. And I remember it very vividly. And really the 2013 versus 2023 meeting was really truly night and day in regards to how far we've come. At the meeting in 2013, I believe there were no industry partners. If there were, they were in the back of the room and didn't speak very much. And I don't think there was really an open dialogue. You know, companies weren't really engaging with us clinicians trying to, you know, help in the uveal melanoma space. And I think really at that time, we were also just starting to understand how uveal melanoma was different from cutaneous. I think we always knew it. But for a long time, uveal was either kind of just completely clumped in with cutaneous melanoma or just completely excluded. And it was really then, you know, starting about 10 years ago that we're like, you know, uveal melanoma needs its own trials and such like that. And then fast forward to 2023, we had 7 industry partners. And like I mentioned, they really represented aspects of primary eye treatment, genetic prognostics, and then treatment for metastatic disease, as well as discussion of adjuvant and neoadjuvant therapies. So these were the sponsors of the meeting, and each of these, with the exception of Castle, actually had an opportunity to come up and talk to us. So I'm not going to belabor too much, because some of this actually will be covered later in the meeting, or in some way, some of the stuff for a lot of the folks that are listening in tonight isn't pertinent because you've already kind of passed this part of your treatment course. But I do think it's exciting, so I will give kind of some of the other updates that may not be as pertinent to the group who's listening in tonight. But Ara was present, and Ara did speak, and they gave an update on AU011, which actually now has a name called Belsar. And this is a virus-like particle conjugated with a a light activated kind of we call cytotoxin to actually kill the tumor. And as you can see by the diagram, this is either injected suprachoroidally, so in that part of the eye, or intravitreally in that, and then actually using a light, it is activated. And really what they presented, and actually what was presented not but a few weeks before this meeting at the American Academy of Ophthalmology, was really some positive data showing safety and efficacy, that perhaps this could be a new primary treatment that actually kills the primary uveal melanoma, but spares patients from dealing with all of the fallout from radiation-induced retinopathy. And the hope is this will be moving into a phase 3 clinical trial. The next company that industry partner that presented to us was Delcath. I will actually talk a bit more about this in my kind of next section coming up. But as many of you know, and perhaps tuned into another webinar that CureM recently hosted, Hepzato is recently FDA-approved liver-directed therapy involving percutaneous hepatic perfusion of the liver. It's available currently at expanded access sites, and hopefully will be commercially available in early 2024. IDEA also was there to give updates. And again, as has been presented at some other webinars, and also will be highlighted, I believe, in Dr. Butler's talk, is that the data in the phase 1/2 has been promising, and the pivotal trial for the combination of darovasertib and crizotinib is now open at multiple sites in the US and abroad. And there are also neoadjuvant and adjuvant trials opening worldwide. Immunocore was also present. We know tebentafusp, or Kimmtrak, is the first FDA-approved therapy for metastatic uveal melanoma. It is now widely available in the US and abroad, and there are some ongoing trials looking at other targets. Replimune is another industry sponsor that was present, who is looking to open a clinical trial in the uveal melanoma space. Their direction is a bit different than ones we've maybe talked about before, and that it's a kind of intralesional, so a direct injection into the tumor, modified virus. So similar to actually a medication called T-VEC, which some of you might have heard about. It is a herpes virus, but it is otherwise modified with something called GM-CSF, and then some other kind of immune tweaks to make it really helpful for the immune system to kind of recognize the cancer. And in a trial of metastatic uveal melanoma patients, they were either given just this injection or in combination with nivolumab. And the response rates were upwards of 28.6%, with a disease control rate of 57%. And so it was really as a result, and you can see some of the characteristics of these patients. So some patients had actually previously already had immune checkpoint, and then ultimately had a response. So it's really with this efficacy that the hope is that this clinical trial will be available to patients hopefully soon. Trisalis also presented on the ongoing PERIO-1 clinical trial, which again, I will talk about kind of in my next session. And this is intrahepatic artery delivery of another type of immune stimulant via a special catheter that allows kind of pressure and prevents backflow in combination with immune checkpoint inhibitors that are given peripherally. And there's been various cohorts, and like I said, I will update some of that data in my next section. And so I think the discussion points from the industry panel after they were done presenting, the general updates, I think we all agreed that it was, that with many more folks interested in developing treatments in uveal melanoma, which obviously is exactly what we've been wanting and asking for years, we really encourage the companies to be aware of the general landscape. So, as a company identifies a drug or a treatment, and they're, rightfully so, very excited about it, when they're writing their clinical trial, to really not have it create competition with other trials. So you can imagine that if, two companies were about to open a first-line clinical trial, we don't want patients to feel like, if they start one, they can't go on the other, or vice versa. So really to not create competing trials. And in the same regard to sometimes make the inclusion criteria or exclusion criteria less restrictive. Again, I always say part of my job for patients is really trying to line up, option A, B, C, D, and E, hoping we stay on option A for a very long time. But if you could have kind of the foresight to say, okay, we'll do E or we'll do A and doing A won't prevent B. But the problem is sometimes when sponsors are writing clinical trials, they want patients who are have never been treated. And part of that is understandable, but the truth is a lot of our patients do get treated with something. So really to make the inclusion exclusion criteria as least restrictive as possible, still obviously understanding that you need to complete the clinical trial and we want to get treatments approved. And then just generally making trials accessible. So opening them in multiple geographic regions, whether that be in the US or Canada or abroad, and really supporting patients so that access to clinical trial is not a reason to not participate. So that was kind of one section of the day, and now I'm going to kind of pivot to another section of the day where we really talked quite a bit about liver-directed therapy. So for many of you who've tuned into previous webinars, you've heard me talk about them, other interventional radiologists talk to them, but we do know that liver-directed therapy is an important treatment approach for uveal melanoma. And when I was developing my talk to give for CureOM at the CureOM meeting, I knew I was talking to obviously a bunch of folks that knew about liver-directed therapy. However, I did think that it was pretty important to just remind ourselves about the pros and cons of really what I consider to be liver-directed therapy versus the pros and cons of systemic therapy versus the pros and cons of combination therapy. Because I think when we think of now that we have more and more clinical or more and more treatment options and clinical trial options, often patients will ask that very question. Well, should I start with liver-directed therapy or systemic therapy? And the problem is what we don't have is clinical trials to necessarily tell us who would be better served by starting with one or the other. So I think at the very least, understanding and reminding ourselves of the pros and cons is very important. So I know I don't have a whole heck of a lot of time, so I'll kind of go through this as quickly as I can. But, the pros of liver-directed therapy, we know that there's a number of various liver-directed treatments that can be very effective in controlling hepatic metastasis. And most liver-directed treatments are not restricted to one population. We do have one specific liver-directed treatment that is FDA approved, which is PHP, but there are a number of others, and some patients may only require limited treatments with long treatment-free intervals. The cons are obviously that it only treats the liver for the majority of patients, and that extrahepatic disease can develop. It requires institutional expertise. Not every even major academic medical center has liver-directed therapy as an option. You really need dedicated interventional radiologists to perform some of these treatments. And some liver-directed treatments do require more extensive resources. So the pros and cons of systemic therapy, we know an effective systemic therapy can treat the whole body. We do have a drug, tebentafusp, with an overall survival benefit with a decent side effect profile. We now have the options of potentially PKC, so darovasertib and crizotinib. But the cons being, we know that tebentafusp is HLA restricted. We know it's weekly. It may not necessarily shrink tumors up front. And then often the systemic trials, like I already mentioned, sometimes are restricted to line of therapy. And then we know the immune checkpoint inhibitors often have a low response rate and high immune-related adverse effects. The pros and combinations of combination therapy, we know that if we could effectively combine them, you could potentially control hepatic and systemic metastasis. There are opportunities to potentially debulk liver tumors before you even start the systemic therapy. And there could also be synergy, take for instance, Trisalis or even like Replimune, there may be this option of synergy of liver-directed treatment with systemic therapies. What we don't know is, should we be combining them at the same time? Should there be a sequence? And then it's obviously managing liver-directed therapy and systemic toxicity. So these are all things we think about when we're thinking about designing liver-directed therapy treatments, trials, and then also obviously making recommendations to patients. So there's a lot of combination type treatments that are underway. My institution at Jefferson, we've been doing immunoembolization for a long time. We've been combining it with immune checkpoint inhibitors over the years. Actually, another group in Seattle is also doing this now and presented recently at SITC. We have PHP followed by checkpoint, the Chopin trial. There's a trial of Y90 followed by checkpoint. SD101 and checkpoint is kind of like combination, like I said, same with the Replimune trial, and then SBRT and LAG3. So very briefly, I do want to just highlight some of the take-home points from TriSalus. This was data that was recently presented actually as a late-breaking abstract at SITC. Like I said, TriSalus is this special catheter that allows infusion of SD101 and also in combination then with immunotherapy. There have been multiple cohorts. And what we really have seen so far is a 58% disease control rate across all SD101 doses, and actually an 81% disease control rate at 2 milligrams. So some of this data is not caught up yet with the more recent patients that we've enrolled. But one of the points that we've been making is that it turns out when we actually treated patients at the much higher dose, it turned out that actually the highest dose may not be the best dose. So we're now actually working our way kind of backwards to try to find the optimal dose. And these are patients who cleared their ctDNA, which is a marker sometimes that we use in addition to kind of radiographic response to decide whether or not a patient is having a response. And then this is the early overall survival signal, which is encouraging, which is not too unsimilar from some of the other trials that we've been showing as of late. Like I said, there's a whole separate webinar on percutaneous hepatic perfusion and this, so I won't go through it too much, but just a reminder, it's really a kind of a catheter system that allows closed circuit perfusion of the liver and infusion of high doses of what's called melphalan, bathes the liver, and then the blood kind of gets sucked out through a filter system, and then blood without the chemotherapy is put back into the body. Data on the results of the FOCUS trial have been presented a couple actually years ago now, in 2022 at ASCO by Dr. Zager. What led to the FDA approval was really this showing that with PHP, there was a statistically significant improvement in overall response and disease control compared to the best alternative care arm. And these are the PFS and overall survival curves. There are some side effects with PHP, mostly related to bone marrow suppression because the drug melphalan, even though most of it gets filtered out, some does stay in the system and it can affect the blood counts. This is just showing that there have been a few trials. I don't highlight here the data from the Chopin trial, but there is a trial that's ongoing right now that shows very promising results in combining hepatic perfusion with immune checkpoint. So here are a couple of schematics that were presented at our meeting, looking at hepatic perfusion followed by immune checkpoint, or actually a dose of checkpoint. Given this is IHP, so this is the isolated hepatic perfusion, which is more of the surgical procedure, followed by immune checkpoint, and then which has then launched into this one now, looking at PHP, which is just really the comparison of systemic immunotherapy versus PHP followed by immune checkpoint inhibitors. And then what I actually presented at the meeting as well is a proposed study design of actually combination tebentafusp and liver-directed therapy. So here is a proposed study design where we would give patients first tebentafusp kind of upfront, get you through the first 3 doses, and then add on liver-directed therapy. And then that would be for patients with kind of lower volume disease, and then patients with higher volume disease actually doing chemoembolization to try to debulk the tumors in the liver, followed by tebentafusp. So these are a couple other combination trial designs that we've been discussing. The next section that I wanted to just mention are adjuvant and neoadjuvant treatments. I'm not going to spend too much time here because, like I said, I think most of the audience is kind of beyond, potentially this stage in your treatment. There was a summary of completed trials. A lot of the data was represented on trials that have completed that you've heard us talk about, like the suit and valproic acid, which was a trial that was done at Jefferson, as well as the adjuvant ipilimumab and nivolumab trial. But the trials to open were also discussed. So a trial of what's called quizinostat, which is an HDAC inhibitor coming out of University of Miami, which has been kind of long awaited to open. The neoadjuvant adjuvant darovasertib trial. So there would be a trial where patients actually with larger eye tumors could get neoadjuvant therapy, followed by definitive eye treatment and then adjuvant therapy. And then actually what was announced also at the meeting was what's called the ATOM trial, which is adjuvant tebentafusp in ocular or uveal melanoma. So again, just really encouraging now that, we have potentially effective treatments in the metastatic setting, that we are now moving them up. Very, very briefly, and we can make these available, just as, Nigel has led us, has already teed up. There are many now active clinical trials specifically for uveal melanoma or have uveal melanoma cohorts. So these are immunotherapy clinical trials. These are targeted therapy clinical trials. These are liver-directed clinical trials. And what I actually went ahead and did was that I gave a talk back in 2018. And there's a thing you can do in clinicaltrials.gov where you can basically map the number of clinical trials available. And this was the map of the US in 2018. This was a map of the US in 2018. This is a map now in 2023. And this is the US in 2023. So just comparing. So there's definitely still more work to be done. But, the fact that there are, more trials in the US, more trials in Canada, more trials in Europe and Asia and Australia abroad, and then just drilling down to the US, the fact that we have states where there's trials that are open that previously were not, this really, I think, speaks to that issue of accessibility, that patients shouldn't have to fly halfway across the country to enroll in a clinical trial. So I thought this was encouraging to look at. I hadn't looked at this in a number of years, but we can certainly do better. And I think that is it for me. So now I'm going to turn it over to Dr. Butler.
Great, thank you. All right, great. So thank you very much. These are my disclosures. So I'm going to review three sections of the meeting. One was tebentafusp in practice. Another is highlights of different systemic therapies. And finally is the vision registry that's part of the CureOM program. So for the tebentafusp in practice, there were three people that presented. I presented some of our Canadian experience. Dr. Sullivan showed some interesting ways to measure responses to tebentafusp, and Dr. Cedar presented some data in real-world data. So tebentafusp is a bispecific molecule that brings T cells to the tumor and has been shown to result in significant survival benefit for patients who receive tebentafusp. And it was approved in 2022 in the US, followed quickly thereafter by the EU, UK, Canada, and Australia. And it's really been extended out across the world where patients are receiving this treatment. But as you know, clinical trials have very strict eligibility where you have to meet certain criteria in order to participate. And it's often to really make the data clean so that the effect of the drug can be demonstrated consistently. And one of the problems with this is that when we treat patients in the real world, especially when a drug is first available, we may have patients who've never had the drug or opportunity to receive the drug who have more advanced disease and or other symptoms that or medical histories that would prevent them from receiving the drug on clinical trial. And Dr. Cedar presented data that or case vignettes of patients with high burden of disease, patients who are actually pretty sick from their cancer, disease involving the brain, or other organ deficiencies like kidney disease or adrenal insufficiency that were excluded from the study. And it basically, the information she showed suggested that there are some situations where patients can benefit from tebentafusp, but it's also challenging, and that perhaps in a patient with a large burden of disease, we need to do something to sort of get the patient more stabilized before starting tebentafusp. And that sort of real world experience is one of the reasons that Dr. Orloff is coming up with an approach with liver-directed therapy prior to tebentafusp to benefit certain patients who are identified later in the course of their disease. In Canada, we initially had a very limited access of the drug in just 2 centers in the country, but now it's being infused across the country, and many different patients are able to receive this treatment. We found that in this real-world grouping of patients that stabilization or response to tebentafusp was associated with better outcomes. And this is pretty similar to what was seen in the clinical trials. So we think that, you know, it wasn't a super specialized group of patients on trial, but actually shows that patients are benefiting. And then Dr. Sullivan presented some novel data to help us to try to think about how to assess patients and their response to treatment. One of the problems with tebentafusp is that it slows down cancer growth, but it may not shrink the cancer. And we typically, as solid tumor medical oncologists, look at tumor shrinkage or lack of growth as a way to say that the drug is helping. If the tumor grows, it's hard for us to know whether it's actually helping or not. It may be slowing down the disease, but we're not sure. There is data that came from the clinical trial. It's actually interesting that the data was generated from samples that weren't actually drawn specifically for the test that was developed. So this is somewhat of a finding that's hypothesis generating, where if you see that circulating tumor DNA decreases, even in patients who have progression of disease on CT scans or MRIs, that decrease in circulating tumor DNA is associated with a better response to therapy. Problem is, the assay that was used in this study isn't really commercially available, and there isn't a perfect test that's been developed for uveal melanoma that we can use today to follow patients. What Dr. Sullivan found is that there actually are several different ways to look at circulating tumor DNA, as well as circulating tumor cells in the blood fraction, and that perhaps one of these technologies will be best suited for patients with uveal melanoma, suggesting even patients who are on standard of care treatment may benefit from clinical trials that use this biomarker as a way to follow patients, and hopefully in the future we can develop new approaches for patients. And then there is a large section. I will only be able to go over some of the highlights where several different systemic therapies were discussed. Dr. Moser showed several slides on emerging therapies. Dr. Hamid's focused on developing technologies beyond bispecifics like tebentafusp. I spoke briefly about protein kinase C inhibition and the darovasertib study, and Dr. Khan also focused in on immune checkpoint inhibitors and some of the current questions that are important for us to answer over the coming years. In terms of the slides that Dr. Moser presented, there are some very interesting early study results looking at PI3 kinase delta inhibitors, which are showing promising results in patients and in animal studies. And hopefully this will develop into another type of targeted therapy that can benefit patients. He also showed a novel concept of using a molecule that brings brings radiation particles directly to tumor to target tumor cells for therapy. And he also presented some of the promising trials that are trying to target tumors with cell therapies. There are actually several different studies looking at TCR-based therapies, CAR T-cell-based therapies, as well as tumor-infiltrating lymphocyte therapies that are being studied across the world, and we're just not sure now what the best approach is going to be for patients. And then Dr. Hamid looked at bispecifics. He highlighted the 3-year survival for tebentafusp, which showed that there's a consistent benefit in terms of overall survival. And it looks like there might be a tail to the curve. Unfortunately, it's not enough patients that are doing really well 5 years later, but certainly they're doing better than they were on the investigator choice protocol. And then he moved on to talk about the new agent that's coming from Immunocore. This is a drug called F106C. It targets PRAME, which is a antigen expressed highly in uveal melanoma, but also in other types of cancers. They've seen actually a high percentage of responses in patients with uveal melanoma as well as skin tumors. And there are several follow-up trials beyond the initial phase 1 to look at the role of this study in patients with uveal melanoma, cutaneous melanoma, as well as other tumors, hopefully in the future. And then he briefly mentioned several approaches looking at T cells that are, or lymphocyte cells that are allogeneic, in other words, cells that come from one person but then used in another person to try to fight the cancer, CAR T cell approaches, as well as other approaches where a bispecific agent would be brought into a cell itself and then infused as a therapy, as well as an approach with something called human mesenchymal stromal cells. So there are various different types of immune cells that are being investigated beyond lymphocytes to try to treat the patient's cancer. And this is the BITE oncolytic virus approach that he briefly touched on. We also talked about protein kinase C inhibition. Darovasertib was presented at ESMO, where darovasertib in combination with crizotinib results in a high response rate in patients who have uveal melanoma. We saw benefit in patients even with large amounts of cancer, although it does seem as though patients with smaller disease burden benefit longer with patients with treatment. And the phase 1 study had a massive accrual over the last year, 2 years, and has now transitioned into a phase 2, phase 3 approach in the first-line setting where darovasertib and crizotinib are being compared to immunotherapy or investigator choice. Well, what will this study do? How will it help us move forward? I think it'll answer questions like how long does this therapy last? Because it'll be a much larger study where we'll be able to compare this treatment to other treatments in patients on the protocol. It'll also help us to understand the side effect profile a bit better in a first-line population, and help us as the community learns how to use this agent, because it'll be administered in a large number of centers, how we can get patients quickly to be able to tolerate the therapy. It does have side effects, but most of us have been able to get patients stabilized on the side effects of these drugs and get them to where they're really thriving with the disease response. And then, unfortunately, what's going to be left after this first-line study is we still won't know exactly how to sequence patients. There will be many questions about whether a patient should be on darovasertib, crizotinib, and certain patients may need to be on this drug right away, whereas other patients we'd like to treat them later on. And I think that some of these questions will need to be answered through clinical trials where investigators will get together with these new tools that will be useful in treating patients, and then we can hopefully come up with identifying who's best to be treated with which sequence of therapy or combination of therapies. I think the other thing that'll be very important is that we need to be able to engage with the pharma and other drug developers to help them understand that patients need more than just first line. They need multiple access to multiple lines of therapy, and that we also need data to help us as clinicians, as well as the payers, to help guide what is the best sequence in therapy so that most of the patients benefit. Dr. Khan reviewed the data on immunotherapy with checkpoint inhibitors. He made the point that while we have a higher response rate with combination immunotherapy, there are many more side effects with combination immunotherapy compared to single agent anti-PD-1 therapy. So when you look at ipilimumab plus nivolumab versus nivolumab or pembrolizumab alone, the response rates are different, side effects are definitely different, but the overall survival in these non-concurrent clinical trials are actually not that different. So while you have a higher response rate, it's very low, still 20% versus 5% for combination versus single agent. But what does that get you in terms of the response rate? Well, it may get us a couple of extra months. There certainly are some patients that do very, very well with either combination or single agent. But as a general population, he pointed out that we're really not sure whether there's a significant difference in overall survival. There's no head-to-head comparison, so it's very hard to really directly compare, but it is important to note. And therefore, immunotherapy-wise, we really need to look at other combinations as possible options for therapy. There is a study that's looked at the LAG-3 combination of nivolumab and relatlimab. Response rate is higher than single agent, but a little bit lower than combination in that study. But it's promising, and it may be an option for patients that want a higher response rate, but maybe less side effects. And then there are also other approaches, such as liver-directed therapy, which Marlana reviewed. There's also exciting results in terms of TIL therapy and other cell therapies that are being investigated. Iovance is hopefully going to have an approval soon for cutaneous melanoma. There are some cohorts being studied with uveal melanoma, as well as other groups are looking at uveal melanoma. And then our own Sarah Seelig presented data on the Vision platform, which is a real-world registry that is actually patient-powered. So rather than researchers putting this together, it's patients and in collaboration with academics to obtain information directly from patients that could then help us to better understand the patient experience. This registry has a incredible objective of collecting data from patients all over the world, and to really get a sense of how there may be geographic barriers and differences in a way patients are treated and the way some of the hurdles that people are dealing with. And I think that this data will really be helpful for us as we advocate for more access to resources for patients. The registry generates a data report once a year, and there are over 400 patients that are registered, and we encourage everyone to participate in this if possible. The registry is looking at data such as how the diagnosis was made, some specific questions regarding patient history and their experience are being collected. I think this is quite important, understanding where barriers to treatment occur, such as financial barriers, as well as what are the priorities for patients. And it does turn out that long-term survival is #1 priority for patients, and how the experience with quality of life, as well as experience with, or the team of doctors taking care of the patient, are important priorities. And I think these things are important for us to understand for patients. So I think Sarah's going to mention that there are some special sessions coming up regarding the Vision Registry, and hopefully you'll be able to participate in that. So in summary, I think we live in an amazing time where there are a lot of new options for patients that previously were not available. There are actual standard of care treatments for patients that they don't have to travel 2,000 miles in some cases for treatment, but can actually get treatment that will help them live longer locally. However, we still need clinical trials to help us understand best how to treat patients. And we also need to advocate so that the regulators understand that this is still a major unmet medical need. I'm a little bit anxious that this mad rush for the first line therapy will leave second line, third line, fourth line kind of in the dust. And some jurisdictions, such as Canada, really the payer wants to make sure that if something works in the first line, it still works in the second line and won't just assume it will. And when that's the case, we really need to generate data to prove that to them, because otherwise they're going to say, well, you get your one shot and then that's it. And I think that ways to do that include participation in clinical trials, as well as collecting real world data. So thank you very much. Happy to answer questions.
Thank you, Dr. Butler and Dr. Orloff. Really appreciate that sort of whirlwind tour through the CureOM science meeting. I want to mention two things about it, and then I'm going to ask Nigel to kind of share again some of his perspective from the meeting and to start the questions off. Just two things, Nigel, before I hand over to you. One is that with all of everything that you both just presented, just to mention that we also had an additional session at the science meeting, which we didn't even cover here, and that was the preclinical session. I just want to mention that, and happy to put something together to report on that session for patients too. So that is research before it gets into the clinical space, and so we thought that presenting kind of the more clinically relevant data right now to patients was sort of the priority. I think the preclinical session was particularly inspiring to me because it's the pipeline, right, for what will hopefully become the clinical component for patients down the road, although it'll take a little longer to get there. So I just wanted to mention that. And the other piece is that I think the other thing that was inspiring at the meeting was that we not only had the presentation of all of these data, but in this room of close to 100 people in the field, we really tried to have discussions about barriers and facilitators to making progress. And so how do we take this information, these data, and how do we really bring it to patients faster? And bringing sort of the minds together of how do we overcome some of these barriers to get to patients faster was, I think, a lot of the discussion in the room that day as well. So Nigel, would love to sort of have you reflect a little bit more and start us off with some questions. And I know we'll probably only have time for a few questions, which we'll try and answer most of them tonight. If you are left with any questions after tonight, feel free to email them in, and we will do our best to post like additional Q&A online afterwards.
Thanks, Sarah, and thank you, Dr. Orloff and Dr. Butler, for two incredible presentations. I have to say, your slides are very impressive. I've been to so many conferences in the past where the presenters are constantly apologizing for their busy slides. Your slides were crystal clear, they were bright, and they popped, and they really help us to understand. And I hope that your work is getting a little happier every day too, with all of these improvements in the field of treatments. Dr. Orloff mentioned neoadjuvant and adjuvant news. You know, that is important to all of us because we have built community over the past 10 years, and what matters to one of us more and more, I think, matters to all of us. Thank you, Dr. Butler, for mentioning the Vision Registry and the Vision platform. That's very important to patients, and we hope that if you have not jumped in with both feet, you will do so after this webinar. This is a chance to tell your story. The oncologists know many stories, but you know your story best. You have lived with it. You're aware of the twists and turns in your journey, what things went well, what things went wrong, how did you get through the rough patches, etc. So watch out for the link to that platform again as we close today. Dr. Orloff, I had not seen the clinical trial map before. I think there may be a stampede towards Texas as a result of it. Let's see what happens. And Sarah, should we start the questions?
Yes, that sounds great.
So let's just, I'll just put it out there, and whichever of you would like to tackle it, please do. Now that there are approved therapies for metastatic ocular melanoma, is there a new role for clinical trials? And how do you help patients understand their options clearly?
Well, I can start. I think that the excitement is that we have treatments that can help patients. The disappointment is that the results are incrementally better than they were in the distant past. That's on top of many decades where there was no progress. So we're making progress, we're excited about it, but we're not there yet. We're not in a situation where the majority of patients are cured. So therefore, I think a clinical trial remains the best option for patients. I remember that ASCO this year, there was a educational session. This is the American Society for Clinical Oncology. There was an educational section on uveal melanoma, and basically someone said, so what do you do if you progress after tebentafusp? And Sophie Pipper-Newman basically said clinical trial, because while we do have treatment, the treatments are disappointing. And if there is a clinical trial that makes sense, and, just any random trial is not great, but if there's a novel trial with a good idea, it's probably your better bet because it'll give you something extra, possibly. And we're not able to do the trials unless patients are interested and committed to participating. And when we're designing these trials, we want to design trials that'll help patients, we're not designing trials where patients will not get effective therapy. I've been doing this for for 15 years or so, and there actually haven't been placebo-controlled trials in many, many years, especially for metastatic disease. Maybe in the for adjuvant setting, there might be such placebo-controlled trials, but almost never will we have placebo-controlled trials.
So time to squeeze one more question in, Sarah?
Yeah, are you looking at the one in the Q&A?
You
Or
go
maybe,
ahead with it.
okay, why don't I take that one, and then Nigel, if there's one more pressing one after. But we have one online here. I would be interested in the results of the adjuvant, neoadjuvant trials that Dr. Orloff mentioned.
And so there really aren't results yet. So the adjuvant, neoadjuvant trials, I don't know specifically if they were talking about the Durvalumab trial. It's ongoing. I think in the chat there was actually a patient who shared that they are participating in that trial, I think in Australia. It is open, will be opening internationally. While there's not data specifically from that trial yet, because it's really fairly new, what I can say is that the trial design and idea came from the fact that there were a number of patients who were enrolled on the trial who had both concurrent primary eye tumor and metastatic disease. And there were some patients who went on with the primary eye tumor intact, also metastatic disease, got treated, and both the metastatic disease and the eye tumor responded. So it was really with that observation that then kind of launched this trial specifically for patients who only have primary eye tumor. Really in an effort, so the endpoint of this trial is really an eye preserving, vision preserving endpoint. So the hope is, could you shrink the tumor enough to change a patient from getting a nucleation to a plaque? Or even if it's a large tumor that would get a large plaque, can you limit the amount of radiation needed, because the plaque size is based on the tumor size, that patients have less of the radiation-induced retinopathy and vision loss down the line. And then there's kind of that second piece where patients also will get adjuvant therapy, and the hopes was obviously preventing metastatic disease. So, hopefully data will read out. As with any adjuvant, neoadjuvant trial, the data sometimes takes more like years to read out. So it may be a little while, but not necessarily, I think, for that first endpoint, which is the eye, kind of the eye endpoint. So keep an eye out for it, and we will certainly update everyone once we have data to share.
And for one,
Karen was asking about the valproic and acid and Sutent studies. I think those studies are still a little immature, right, Marlana?
Oh, sorry. Yeah, I didn't highlight those because actually a lot of the data was actually presented at our patient meeting almost a year ago, August. There was some updated data presented on that last cohort. Essentially, it looks like Sutent for 12 months probably does best compared to historic controls, which did not get any treatment. But the last cohort, the data is still kind of maturing. But I would say, is Sutent going to move forward as like an adjuvant therapy option? I think it's always a challenge, in cancer drug development. The normal protocol is a drug that's effective in the metastatic setting gets kind of moved up and adopted in the adjuvant setting. When we designed the Sutent valproic acid trials now many years ago, there were no effective metastatic treatments. So I'm not certain Sutent valproic acid, even with what looks like some potentially positive data, is going to kind of go the distance the way some of the drugs that are showing efficacy in the metastatic setting that we're now testing in the adjuvant setting might.
Thank you, Dr. Orloff. Nigel, do you want to close us out with the last question?
yes, and maybe it's a forward-looking question again for either of you. if patients wanted to believe that there is a trend to longer survival for metastatic ocular melanoma patients, where would they look for evidence or data about that?
I think one of the reasons why the registries are important is that this will include everyone, that not just patients that fit the requirements of a clinical trial. There are actually, in addition to the Vision Registry, there's also a registry that's coming from the cancer centers that's physician-led, and I think this will include patients who have a diagnosis and then we follow them. We won't have the same level of quality of life and other sort of some of the the data collected will be a little bit different in the different registries, but they'll be complementary. And then when you look at clinical trials, patients overall are doing better on the clinical trials that are currently happening. Anthony Joshua did a study where he looked at all prior clinical trials prior to around 2010, and then looked at the overall survival for patients in second-line trials or first-line trials and multiple trials, and saw that there wasn't a very high survival rate, and we're already going better than that as well. So I think those are areas that are very suggestive that we're seeing improvement in survival.
Dr. Orloff, did you want to add to that?
No. Yep. No. I think like Dr. Butler said, I think you look at any of the recent trials and we look at a 1-year overall survival, 50, 60, 70+%. Whereas, what Dr. Butler mentioned was what they call kind of the Rentala kind of paper, which looked at all patients on clinical trials a number of years ago. And when you compare it to that survival, we are doing much, much better. We can do better, and I think to go back to your point, why clinical trials? And I think the bottom line is because we haven't found a cure yet. We need clinical trials to keep moving the needle. And so, little by little.
Well, I think that's a good transition and actually brings us back to the origin. Cure OM is to find the cure, and I know everyone shares that goal tonight on this webinar. So I really want to thank everyone for joining and participating, and I think these are great questions. And again, we can continue the Q&A. So if you have pressing questions that didn't get answered tonight, feel free to engage with us, email us, and we're happy to work to get additional questions answered. So with that, I'd like to thank Dr. Orloff, Dr. Butler, Nigel, Deacon for updating the patient community about the research and clinical trials that are taking place in the OM community and what we were able to discuss recently at the Cure OM Global Science Meeting. As I've said multiple times, we are grateful to you all and appreciate all of you for what you're doing with and for the ocular melanoma community around the world. Appreciate your time this evening. And just a reminder to everybody, this program will be available on demand to listen to later, or you can share it with others. Miriam, I think you've put a link in the chat, or folks will receive that afterwards. And also a reminder of our support groups. So just to make sure you're aware of our upcoming support groups that run the first Wednesday of the month and the fourth Friday of the month. And if you forget that, or for any other reason, please feel free to reach out to us at cureom@melanoma.org for more information. Again, to reiterate what Miriam said, we'll send out an evaluation after this. We really value your feedback, and don't hesitate to reach out to us for any reason at any time. And we look forward to additional times and spaces to learn together and to support each other. So thank you, everyone.